Hello, I'm Juan Pablo Llopart. I'm from Uruguay and I am here to present this study, which is part of my great thesis titled The Effect of the Invasive Argentine Ant Venom in Amphibian of its Native Range, in which I work with Paloma Álvarez, Lucía Moreira de Marco, Alok Bang, Elena Ángulo, and Raúl Maneiro. 𝒉𝒖𝒎💊💕🉬 humile is among the 5 invasive ant species. As you can see, it is present in almost all continents. They attack in large numbers and don't need a stinger to defend themselves, because they spray a potent venom effectively against competitors and predators in areas it successfully invades. It has been shown the potential of the venom of the Argentine ant to kill amphibians in the invaded ranges, so they affect all levels of the food chain. Here we propose that this hypothesis, novel weapon hypothesis, could explain the successful invasion of the Argentine ant, since its venom can be lethal in juvenile amphibians in the area successfully invaded. The novel weapon hypothesis assumes that the weapon exists in the species in native range but does not impact coexisting species. One mechanism to explain this fact is coevolution, such that that species in the native range could have coevolved and adapted to the weapon. Therefore, we examined the effect of the Argentine ant venom on amphibians coexisting with the ant in its native range, because amphibians with different degrees of myrmecophagy could have coevolved and adapted to prey's venom differentially. Rhinella arenarum presents a high number of ants in its diet, Odontophrynus americanus as an intermediate degree, and Boana pulchella presents the smallest number in its diet. We evaluate the venom's immediate effect after exposure, monitored the short and medium-term effects, and discuss the results along with the venom impacts in invaded ranges. On-field amphibian species were collected in larval stage and reared in the laboratory until metamorphosis, after which they were placed in individual conditioned containers. Tadpoles were fed ad libitum with boiled lettuce and reared in aquariums. Ants were also collected and maintained in laboratory for subsequent experiments. When the larvae reached the metamorphic stage, the experiments began. During these experiments, the amphibians were exposed to the venom, and clinical studies were conducted to observe their condition immediately after exposure, 10 minutes post-exposure, 1 hour post-exposure, and 24 hours post-exposure. Subsequently, some individuals were selected for a 14-day follow-up to monitor their survival. Now I will explain precisely how the experiment and clinical studies were conducted. For the experiments, approximately 20 individuals of each species were taken, of which 5 were randomly selected to serve as controls. The exposure to the venom consists of a macerate of ants with the dosage calculated based on a ratio of ants per gram of toad. The venom was applied to the back of the toad, as shown in the image here. In contrast, the controls were given with given water to simulate the handling of the individuals. The individuals were then evaluated and classified as affected or unaffected based on the condition of the following response and reflex. If at least one of these responses was absent, the individual was classified as affected. In our results, we found that all three species were affected by the venom, as seen in the dose-response curve here. The most myrmecophagous species, Rhinella renarum, in red, was the most resistant, requiring a minimum of 225 ants per gram of amphibian. The venom take effect. In 𝒊𝒏𝒆𝒑𝒊𝒕𝒉𝒆𝒎𝒂, 171 ants per gram of amphibian were needed, and in 𝒊𝒐𝒊𝒕𝒉𝒆𝒎𝒂, 108 ants per gram of amphibian were needed. To observe the short-term changes, we referred to this flowchart. It first shows the number of individuals used from each species here. Within the treatment group, you can see how the amphibians recover over the course of 24 hours here. The number of affected is decreasing while the number of recovering is increasing here. The unaffected here and the affected here. Compared to European species, we see that amphibians from the native range of the Argentine ant showed different toxic dose values than amphibians from the invasive range. Rhinella renarum and 𝒊𝒐𝒊𝒕𝒉𝒆𝒎𝒂, these two, were more resistant than their European counterparts, 𝒉𝒖𝒎𝒊𝒏𝒆𝒂, However, this prediction did not hold true for the European 𝒉𝒖𝒎 meridionalis, which for some reason was much more resistant than the other species. Then here we can see the 14 days survival, where all individuals of 𝒊𝒐𝒊𝒕𝒉𝒆𝒎𝒂 survive. However, qualitatively, the treated individuals grew less than the controls, as you can see in this growth graph. On the other hand, Rhinella renarum decreased to 60% within the first 2 days, and in 𝒊𝒏𝒆𝒂 survival dropped to 20% within the first 4 days, showing the worst medium-term results. Therefore, we conclude that the venom of Argentine ant affects the studied amphibian species, demonstrating its low specificity. As we observe, it affects both native and invasive species. In the native species, both short-term and medium-term effects were observed. This still shows the potential magnitude of the negative impacts of this novel weapon for already threatened taxa, the amphibian, of course. As 𝒍𝒐 calamita and 𝒊𝒏𝒆𝒂 turned out to be more sensitive to the venom than the American counterparts. Location with threatened amphibians must be highlighted and prioritized for management, as the impact of the invader should not be underestimated. Here are the references for the presentation in case anyone is interested. And now I will open the floor for questions and provide my contact information for any further thoughts. Bye.